Alright, so I'm going to discuss cell communication here with a little video using these props. So basically I'm going to go over G protein linked receptors and receptor tyrosine kinases to show two different types of cell reception and how that can go on different cell transduction pathways and have a different cellular response. Okay, so basically we're going to start out here with some pens, and so we start out with the G protein linked receptors. And so if our cell is the table here, this receptor will be on the it will be located inside the membrane because it is hydrophilic, the molecules that are trying to get in. So we have this will be our G protein linked receptor, and so this candle right here. So we have our ligand, which is going to be this red pen right here. Ligand will attach to the G protein linked receptor, and then that will activate the G protein to come over, which is going to be this green pen right here. It's going to come over and it's going to attach to the activated receptor. And so then this candle right here is our G protein linked receptor, and that becomes activated. And so basically what happens is a GTP molecule, which is this ballerina right here, it's going to come up. It's going to give a phosphate group to the G protein. Alright, and so our phosphate group is going to be this little blue thing. And so that's going to give the phosphate group to the G protein, which is going to activate it. Once it's activated, it can dissociate into another enzyme, which is going to be this crystal bar right here. It goes here, it's activated, that enzyme becomes activated, and that can go on carry out another cellular response, which will end up being, let's say we have another protein here, and that's going to send the signal to make the rocking horse rock. There we go. Alright, so that's the first one. So let's put all this back. That's the G protein linked receptors. The second one we have is the receptor tyrosine kinases, and so we're gonna have these two black Sharpies. Actually, let's get this one. So we have these two black Sharpies, and basically what happens is these are both receptor tyrosine kinase proteins. So we have two ligand molecules, which is going to be these two different types of Sharpies. Once they attach, they are going to dimerize, and they're going to come closer together, undergo dimerization, and then basically what happens is dimerization, it forms this dimer, and then that means it's activated, and so what happens is the ATP comes in once again, and it phosphorylates and gives a 6 phosphate ions to the 3 tyrosines. So let's just pretend it's giving it phosphate ions here. And so this is all going inside the cell membrane too. And then when that happens, different relay proteins will come, such as these two pencils, and attach to the phosphates. Let's make this yellow, the phosphates. It will attach, and then it will carry out two different cellular responses. One could be, let's say, lighting the candle, and then the other could be rocking the rocking horse. And that all happens because the phosphate groups have these other relay molecules, or relay proteins, come over and it activates them, which helps carry out the two cellular responses. Alright. Thanks.